All right, good morning, everybody. Let's get cracking. So before we move on to the next subject, which is transposons, someone asked me, I can't remember who it was, maybe Caitlin, if I tried to find some pictures of that chromosomal territories stuff. And so after class, I kind of did some reading and found a nice paper which is linked here. I've actually uploaded this newer version of the PowerPoint to Blackboard as well. And so this on the right is actually it's a merged picture. And so I'm pretty sure they're using a technique called FISH, which stands for fluorescence in situ hybridization. Basically, they're using DNA probes, from what I remember. I've never done that technique myself, but they use DNA probes which are labeled with different fluorophores, which emit light at different wavelengths. And so the top picture is essentially a composite, right? So it's lots and lots of pictures taken at different wavelengths. You know, wavelength of green, wavelength of blue. Oh, you're welcome, Kaylin. And so on. And they're all kind of merged together. And this is what a nucleus of, I think it's a fibroblast, human fibroblast. So, oh shoot, what are those involved in? Bones? No, those are osteoclasts. I don't know. Fibroblasts. Connective tissue, I think. Let's go with that. And so when you stack all those pictures together, you get this composite, which is what a nucleus actually looks like if you can visualize all of the individual chromosomes. And so they kind of basically made together this map, and you can actually see, you know, here's the X and the Y, two chromosome ones. Obviously, there's going to be other chromosomes on the other side because it's a three-dimensional image. Let's see if I still have. Badges, I closed that tab. Give me just a second. Let's see if we can go find it again. There we are. Oh yeah, this image. There's a whole bunch of stuff in there that I don't understand, to be honest. But so these are all the different images. DAPI stains DNA, and there you go. These are all the different. CY is like a cyan, so these are different wavelengths of cyan. So that would be like this kind of off green color. And the cool thing is because these are taken with a laser scanning microscope with all kinds of funky stuff. This is right at the bleeding edge of microscopy and resolution. That's why this image, well, that image looks so fuzzy, right? Because it is really at the bleeding edge. But what you can do is you can actually make a 3D model. And so you can rotate the image of the nucleus around to look at different parts of it, which I always think is super neat. I've got some images like that too. And this is where they actually model the individual chromosomes using all kinds of funky computational stuff. So yeah, I think it's cool. I mean, I'm not sure I'd be able to do this kind of work because this is really, really advanced microscopy. They're working at really, you know, really tiny scales, right on the edge of what a light microscope is able to see or resolve. Actually, is more the issue. But anyway, I thought that was fun. So thanks for the question. That got me wandering around the interwebs, finding stuff. Anyway, so that's in the PowerPoint for your pleasure. Thought it was cool to add, you know, just a little bit extra. So what we're going to move on to today is actually something that is something I've worked on. So I've done a bunch of things over the years, but one of the projects I worked on was using, or the aim was to use transposons to make transgenic parasites. This is actually sounds a little bit far-fetched, I know, to deliver a biocontrol agent to possums. Because I worked in New Zealand and Australia for some years, and possums, not the ugly ass opossums that you have here, they're actually very pretty. Let's see if I can show you a picture of one. What are they called? Trichocereus vulpecula. I don't want any skulls. Oh, Trichuris, fine. Nope, that's a parasite. Don't want that. Dang it. There you go, common brush-tailed possum. So yeah, that's what they look like. Aren't they cute? They're nasty ass little individuals, that's for sure. I mean, obviously, because we're sticking them in a cage and like jabbing them and stuff. I actually got bitten by one once, which was not a lot of fun. Not even a little bit. But anyway, these things are a plague in New Zealand because they were introduced back in the 1800s to found a fur trade because their fur is fabulous. Obviously, it's the best part of them, ideally without possum inside it. But their fur is fabulous. So possum fur merino wool is actually, oh, I mean, I guess the possums aren't that bad, but I think they're kind of ugly, but that's just me. Anyway, these were released from Australia, which is where they're native, to New Zealand, where they're not, to make a fur trade, basically. Actually, any kind of possum merino mix you can find is absolutely fabulous. And they exploded in numbers. There are, I think, on best estimate, something like 50 million of them in New Zealand. And they just eat anything and everything. Actually, a guy I worked with, he used to, for a fun evening, he used to go and just shoot them, you know, with a 2.2 rifle or something like that. He'd just go around at night with a night scope and just plonk them off the trees. And supposedly in some parts of like more rural New Zealand, you get a beer per possum tail. So if you go into like a local pub and you put down like 3 possum tails on the bar, you get 3 beers, which is good to know, I guess. Anyway, so my job was to try and create a vector for a biocontrol agent that would basically sterilize these little bastards. And I don't actually know what happened to that job because I left before it finished. It didn't go very well. It's an interesting place, New Zealand. So what they do now is they just kind of firebomb the place with really nasty poison instead. They're really crazy. They produce a lot of little babies per year. I think on the South Island, New Zealand, they did a study where they carpeted like a whole area of probably about the size of, I don't know, Alabama, something like that, with poison, wiped out every single possum. Within a year, the numbers are exactly the same again. Yeah, anyway, so that's a bit of a tangent, but it's just to kind of introduce the fact that transposons are pretty funky things. And they're actually really cool as well. So they're a very useful tool, at least before CRISPR-Cas9 came along. And they still are to some extent. But also they're really cool because they are kind of one of my favorite sorts of scientific discoveries, which basically breaks dogmas. So I'm a bit of a rebel without a cause when it comes to science, as you probably guessed. I don't know. But anyway, and so I really like things which kind of turn our understanding of biology of the world on its head, right? Because those are just fabulously interesting. Like the whole Helicobacter pylori and cause of stomach ulcers, right? So that's absolutely fabulous kind of one as well. But this was back in the 60s, 70s. where there's a researcher, a lady by the name of Barbara McClintock. Let's see if I can spell that. I think she's still doing science too. She is an absolute legend. Literally. That's her name. I think I spelled it right, something like that. But anyway, she was studying the inheritance of traits in maize, you know, stuff you make tortillas with. And she kept seeing this weird inheritance pattern which didn't fit any Mendelian patterns, right? And essentially what was happening is that she saw changes in pigmentation, right? Similar to the kind of stuff that you've seen in the corn genetics lab. And it wasn't inherited in a Mendelian way, right? So she really struggled to kind of figure out what was going on, right? How to explain this stuff. And actually, many of the most interesting scientific discoveries, in my mind at least, are ones that are accidental. You know, you see something weird and you just don't dismiss it. You go, ooh, that's interesting. I wonder what's going on here. And that actually often leads to some of the coolest discoveries in science. The whole penicillin, discovery of penicillin, or the penicillium fungus, I guess, yeah, is one of them, for example. Anyway, so came to the end that the only explanation that she could think of is that there were mobile genetic elements that were randomly moving around in the genome of this maize plant. And every so often they would inactivate a pigmentation gene, or a gene involved in pigmentation. And so that would give a kind of spotty color where that pigment gene was knocked out. But because that could just happen randomly, it wasn't a Mendelian inheritance or Mendelian trait. And she was widely panned for this theory. You know, she was like, oh, a bunch of like, this can't happen. But she was clearly right. And she did some really good work to show that. And she got a Nobel Prize for it too. That's how kind of momentous of a discovery it was. Anyway, so essentially what transposons or transposable elements are small pieces of DNA, and they typically range from a few thousand to a few tens of thousand base pairs, right? So they're not huge in terms of like chromosomes, and they are in both pretty much every living organism. You have a whole ton of them in you, for example, in your genome. At least almost half of your genome is transposons. And they encode the ability to move themselves around, right? So these are independent pieces of DNA that move around without any control from the host cell, the host organism, to some extent, right? We do try and dampen it down because transposons are highly mutagenic. So they are hugely complex in terms of different types. I mean, they're not complex in and of themselves, but there's a huge range of different types of transposons, size, kind of genes they encode. And they're one of the reasons why we have a large genome, right? Remember we were talking about the C-value paradox or enigma? Transposons are one of the reasons for that, right? And so they are able to move around and insert and cut themselves out, copy themselves in a way that is very much distinct from homologous recombination, right? And we'll talk about how this works in a second. And so their main mechanism, by and large, is causing a double-stranded break. So all transposases, which is the enzyme that I guess mediates transposition or movement of the transposon. make a staggered double-stranded break. And so what I mean by a staggered break is that if you have your double helix like so, right, homologous recombination breaks it. So you have a blunt-ended break, right? And literally called blunt ends. Now, transposases or transposons are different. What they actually do is make a staggered break. So what you end up with is a bit of overlapping single-stranded DNA on either end of the break. And they recognize a particular DNA sequence. So for example, the transposon that I worked on, which is called piggyback, recognizes AATT. Right, so anywhere that you have an AATT in your genome, if you had a piggyback transposon there, would serve as a site. So the site is specific to the transposon. Some of them have longer recognition sites. Some of them have shorter ones, right? Obviously, the shorter the site, the more frequent you'll find it just by random chance in the genome. So the more potential sites per transposition there are. Right. Anyway, so once the target DNA has been recognized and found by the transposase, it cuts it and it creates that staggered end. And that's a really important characteristic because it causes a pretty major consequence once the transposon has gone in. So once the that double-stranded break has been made, now the transposon comes in and it gets stuck in between that double-stranded break. Right, so one of the overhangs is stuck using the transposase to one end of one strand of the transposon, and the same for the other strand. Now, what this means is that there's going to be two small stretches of single-stranded DNA, right? And essentially what that happens is that these get filled in, right, by DNA polymerase that's involved in DNA repair. And when that happens, you get these things called flanking direct repeats. So these flanking direct repeats are essentially duplicates of part of the target sequence. And these essentially result in what's called a transposon footprint. So that if you're scanning through a genome bioinformatically, right, looking at the sequence information from a genome, every time you see that little doublet, even if the transposon is no longer there, that tells you that a transposon was once there, right? So even when that transposon leaves, right, if it's a cut and paste transposon that jumps around, they're also called jumping genes, actually, is another kind of colloquial name for transposons. When it jumps out, it leaves those direct repeats behind. And so even if a transposon is not there, it still generates mutations in its host genome. Galen, it is always mutagenic. So transposons always cause mutations. Whether they stay, basically because there's a large bit of, I don't know, large bit of foreign DNA present, or if they leave, because you have the flank indirect repeats stay behind. So whether or not that causes problems, though, depends very much on where that transposon has transposed, right? Where it is gone or where it's left from. So if it's in the, I don't know, let's say 90% plus of your genome, which doesn't have a known function, you know, who cares? I mean, there's a reason why there's a bunch of like missile testing ranges in Utah and New Mexico and stuff. Like there's nothing there, so no one cares about, you know, blowing up a bit of the desert, basically. So if that transposition occurs in, you know, part of your genomic wilderness, then it has absolutely no effect whatsoever. What do you think would happen if this transposition occurred in the middle of a protein coding region? So in the middle of a bit of DNA that encoded a protein, what do you think would be the consequence now? Do you think that would be positive, negative, or neutral? Negative, right. Why do you think that would be negative, Isaiah? Why do you think that would be a bad thing? What would be the consequence of having a large lump of foreign DNA in the middle of a protein coding region? Are you still going to get a protein produced, or a functional protein at least? Yeah, you get a mutation, Isaiah, that's completely correct. But one thing that I want to get across is that the impact of mutations varies depending on where the mutations are. So protein coding regions, if you throw in a large chunk of, actually I almost think of transposons as parasites, to be honest, like DNA parasites. If you throw in a large chunk of DNA, you're going to disrupt the protein coding region. You're not going to get a functional protein. So it will be negative, but it'll be negative because you're getting rid of function. Right. Now, interestingly enough, because I've, you know, done some reading recently on, just by chance, on transposons, they actually think that transposons have driven a lot of increases in fitness or novelty, right? Because if one of these inserts into a promoter region, right, so a region that controls the expression of a gene, then potentially you can bring new function, right? So these are basically these regions within transposon are DNA binding domains, right? Or they're recognized by proteins. And so they actually think that transposons have introduced, you know, basically novelty, right? How would one put it? New functions for existing genes, which if those confer a fitness advantage, can therefore lead to, I don't know, Caitlin. All sorts of funky stuff happens in animal development. I actually saw on our porch, a small Mediterranean house gecko with 2 tails. And so I don't actually know what caused that, but I know in salamanders and frogs, particularly frogs, if they get a parasite which ends up in a part of their body as a tadpole, that actually can lead to a bifurcation of one of their limb buds, and so they end up with 2 limbs. I don't know about that, because that's different. That's autotomy. So normally, if a lizard's tail breaks off and it regrows, you get like this kind of smooth, not quite like its original tail grow back. But this was actually two tails growing from, so it might have been potentially a break where the existing tail kind of hung on and then a new one kind of grew out. I don't know, but there's all sorts of weird and wacky stuff that happens. So as for an alligator with a dolphin type tail, it's quite possible, right? You know, it just depends on what it was that changed the development of that particular part of that particular alligator. So whether or not it's down to a transposon, I don't know. I mean, possibly. I don't really know, to be honest. And one would have to look into that in quite some detail. One would actually have to know how alligators develop their tails in the first place to be able to figure out how that might have changed to generate something like a different tail shape. But quite possible. I mean, it could have been a mutation in some developmental pathway. Whether it's caused by a transposon or not is a different matter. But yeah, who knows? I mean, hopefully we're not going to have like alligator dolphin hybrids, you know, jumping around and snatching puppies in the Everglades and stuff. That would be a little bit scary. So anyway, the end result of transposition is a transposon in a new site, right, which didn't have one before, and these two flanking direct repeats. And so when you look at a structure of a transposon, right, so here are the flanking direct repeats on either side, right? You also, in terms of its like what actually it looks like, it has a region in the middle, and that region encodes for a transposase, right? So it encodes the enzyme that moves itself around. That's why they're like these independent bits of DNA. And they also have what are called terminal inverted repeats. These are, and they're basically they're repeats that are mirror images of each other, right? So they point out kind of in the opposite directions. And those are recognized by the transposase to kind of cut out the transposon and stick it somewhere else. Now, there are many, 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 many different types of transposons. There are DNA transposons, right, which are more common in prokaryotes. They can either go replicative transposition, right, so they copy themselves, or they can undergo what's called cut and paste replication, where you cut out transposon, you leave behind the footprint, and then you stick it somewhere else, creating a new set of flanking direct repeats. There are also kind of really weird one called retrotransposons, right? And so retro obviously in popular culture, it means like, you know, wearing like really tight trousers with flares or something like that. Or I don't even know what is considered retro anymore. I know, like those really high waist jeans that they used to wear in the 80s. Anyway, I'm not much of a fashion guru, as you could probably tell. But anyway, so retro in terms of molecular biology means turning something from RNA to DNA. Or not something, information, I guess. So a good example of that obviously is a retrovirus, right? So retrovirus, e.g. HIV is the most famous one. There are others. Varicella, I think. Yeah, I think that's an RNA virus. It causes shingles. Hopefully none of you have shingles, but my grandmother used to have it, and it was an absolute bear for her. And basically what happens is, and this is also why HIV is very hard to eradicate in an individual, basically it's because they're RNA viruses that turn their genome into DNA and then integrate into their host genome. So people with shingles or HIV have that virus forever, essentially, in their genome. And typically, it gets reactivated under particular conditions. What those might be vary from disease to disease. I think shingles is like if you get like really run down, particularly in winter, at least in colder climates, it kind of flares back up again. So it basically comes out of your genome again and starts replicating. So both these and retrotransposons use an enzyme called reverse transcriptase. And that's the enzyme that uses RNA to make DNA. Super useful one in molecular biology, actually. I've used it a whole bunch. If you want to study RNA, like messenger RNA, transcription, stuff like that, one of the first things you do is you turn that into DNA, because DNA is stable, it hangs around. RNA is not. And actually, again, another dogma breaking, right? So the central dogma, actually is, which we'll cover next, a lot of people think the central dogma is about DNA to RNA to protein. It's actually not. This is like my own little personal crusade. It's actually the central dogma is that you can't go from protein to mRNA or mRNA to DNA. The other way around, the one that everyone calls the central dogma is actually called the sequence hypothesis. Anyway, so that was a big kind of part of molecular biology from Watson and Crick's time. And reverse transcriptase obviously broke that dogma because it shows you can go from RNA to DNA. And these are pretty funky, to be truthful. And also you can see why we have such a huge amount of transposons or DNA encoding transposons in our genome, because these copy themselves. So it's not a transposon jumping around, it's a transposon staying still, making another copy of itself that then goes and inserts somewhere else. And so they do that by transcribing themselves, right? So using host machinery to make an RNA copy of transposon. And so that's with terminal inverted repeats, the bit of the DNA encoding whatever's necessary, like the reverse transcriptase and the transposase. That reverse transcription makes double-stranded DNA, and we don't have reverse transcriptase normally in our genome, right? So anything, whether it's a retrotransposon or a virus that uses it, has to bring it itself, right? I think reverse transcriptase is actually a target of one of the drugs that controls HIV, because it's an ideal target because we don't have it. So you don't have to worry about toxicity or anything like that. Anyway, so it makes double-stranded DNA, and that then is the thing that's inserted into the double-stranded break. So you first make a double-stranded DNA copy of that transposon, and then with the transposase that is encoded by the transposon, you undergo the same process up here where you have a double-stranded break. It gets ligated into the break, and then these flanking direct repeats are created. So the last part of the mechanism is the same. It's how that new copy of the transposon is generated that's different. That's the retro part of the retrotransposon. Now, one thing, oh, flyboys are out and about. One thing that's actually really kind of interesting as well is that another reason why we have such a large number of transposons kind of in our genome, it's not just because they can copy themselves, it's also because one, they can actually die. Now, so you're not going to shed a tear for these, but if there's a mutation, just a naturally occurring mutation, as your genome is copied during DNA replication or some other cause, right? Because these are copied with your DNA, right? So your offspring inherit all of the transposons that you have, right? And they may in turn have a few more kind of themselves. But if there's a mutation in this region, in really any part of it, right, particularly the terminal inverted repeats, so if this sequence changes, the transposon will not recognize it. The transposase, sorry. And so it cannot move. If the transposase, the actual protein coding region for the enzyme that moves it, if that has a loss of function mutation, again, that transposon is dead. Unless there's a related transposon nearby that produces that transposase for this transposon to use. So when these accumulate mutations, right, and these mutations are kind of neutral from the perspective of the host, then that transposon is functionally dead, right? And so you can see how, you know, that kind of term that I use, that evolutionary cruft, right? You know, the kind of evolutionary baggage that we accumulate over evolutionary time as a species. You can see how we kind of accumulate more and more baggage over time. And so, as I mentioned earlier, transposition is always mutagenic. And it's actually so mutagenic that many cells have evolved mechanisms. I don't actually know what these mechanisms are. I'll have a look into it just because I don't know. It'll be interesting. To limit transposition, right? Because it is so mutagenic, right? And Drosophila is actually one of the organisms that has been studied the most in terms of transposons. And that's where Piggyback comes from, the one that I was working on. And so over half of all spontaneous mutations in Drosophila are due to transposons. They are really incredibly mutagenic. And another interesting point as well is because these copies, right, unless random mutations accumulate, these copies are identical. Right, so if you have, let's say, one of these copies in one chromosome, say chromosome 2, and a copy in chromosome 4, then potentially during meiosis, remember when those chromosomes kind of align very precisely, right, and recombine during, you know, by homologous recombination, potentially, if you have the same transposon on both chromosomes, now you can have a different chromosome come in and line up and undergo homologous recombination and generate a completely different chromosomal fusion. And so it's actually thought that a lot of the large scale evolution in terms of our chromosomes. So I know that in chimpanzees, they have a chromosomal fusion. Can't remember between which two. So in humans, Homo sapiens have 2 different chromosomes. Can't remember what numbers they are. In chimpanzees, they've actually been merged into 1 chromosome. And it's thought that part of the way in which that happens is through transposons creating these identical bits of DNA in different chromosomes. And also you can get chromosomal duplication where you get recombination between sites of the same chromosome as well. So they are very funky things, very funky indeed. And they're also very, very useful in terms of, or at least until CRISPR-Cas9 came along, of molecular biology, because essentially what I was trying to do was you can cut out the center of a transposon using, you know, molecular biology techniques. And as long as you can produce the transposase separately, maybe even just purifying it and injecting like liquid protein, right? Like the protein that you used in proteinase K digest or Taq DNA polymerase, actually injecting that, that transposase will move whatever transposable element you create into that organism. So you can cut out the center of that transposon and stuff what you want into it. And so that allows you to move a bit of foreign DNA, truly foreign. Like in my case, I was trying to use a GFP reporter, right? Green fluorescent protein reporter. I wonder if I can show you some pictures of that. So transposons consist of essentially the flanking, well, not flanking, the terminal inverted repeats. Those are the bits that are recognized by the transposase, and a coding region for whatever proteins are needed for transposition. And so in the case of simple ones like piggyback, no, so these are not colored. There's a lot of these pictures. Let's see if I find a nice looking one. So in the case of piggyback, it's just transposase gene, terminal inverted repeats. That's it. It's just a small piece of DNA. I think it's around 3,000 base pairs, 3 kilobases. So what I was trying to do is cut out the bit in the middle that contained the transposase and stuff in instead a protein which fluoresces green when you hit it with blue light. And so this is what that protein looks like in a worm. So you can see it's expressed. This is the intestine of a worm, and these are the nuclei and the dang it, what's the name of the real small thing in the nucleus? I can't remember off the top of my head. Nucleolus, I think. That's a nucleolus, and this is the nucleus. And all of these sparkly bits are actually punctate bits of protein. And so my aim was to basically use that as, let's see if I can get some nicer pictures. Oh no, that's CDK. Don't want that. Anyway, I can't find those easily. Was to take that gene and stick it into a transposon, stick the whole bit of DNA like a plasmid, basically a little cargo bit of DNA, controlling my new transposon, right, which is basically a foreign gene with the terminal inverted repeats on either side, and provide the transposase myself. And the hope was that that bit of DNA would then jump out of the carrier and jump into the organism that I was working with. Never got it to work, unfortunately, for a bunch of different reasons. So you can basically put whatever cargo in there, as long as you can provide the activity of the transposase separately. And so that's how you can use this as a tool for molecular biology. And originally, like eventually, the aim was to put the bit of DNA encoding a, I don't know, I think it was a, oh, what was it called? Gonadotropin releasing hormone. It's basically a hormone involved in ovulation in possums, actually in humans as well. And so if you put that into there, then the aim was to get that into a parasite and then sprinkle parasites over New Zealand, basically. And though once that parasite got into a possum and infected it, it would sterilize the possum. Sounds a bit science fiction, but that was what I worked on for a good couple of years, many moons ago. So yeah, they have code. They encode the means to move themselves, whether it's transposase or transposase and reverse transcriptase. And when they transpose, they create these flanking direct repeats. And the reason why they create those flanking direct repeats is because of these staggered cuts that get filled in. And so you can see the overhang here, if you kind of zoom in, right? The overhang here is TCGAT, right? It's just what's created by the transposase when it cuts the DNA. And so because that gap exists over here, now you have a second TCAG made, which is a copy of the first one, this overhang here. And so that's what creates direct flanking repeats. So yeah, there you go. That's transposons. They are pretty crazy. So next up we have, really we're going to start on, I hate calling it central dogma for obvious reasons, I've already told you, start on gene expression. And that's going to take us all the way to exam 3. Actually, I think that's Guy Fawkes Day in the UK. There you go, a little bit of culture. And where we're going to go into, really, and there's a lot of details involved in this, how you turn the information in DNA into something functional, typically protein, right? So that's what we're going to spend the rest of this section on. And then the last section, section 4, will be how that's controlled. That's going to be the bulk of it. And if we get time, talk about some of this fun biotechnology transgenic stuff. Cool. That's it from me. Does anybody have any questions? All right. Well, no point at the moment. No point going on to the next subject. We'll start that on Friday. So I'll see some of you in a little bit for lab, and the rest of you have a lovely day, and I'll see you on Friday.